at the Barbara Bush Children's Hospital in Pediatrics, and she completed her bachelor's degree in science in biology, Bachelor of Science in Biology, graduating magna cum laude from the University of Massachusetts at Amherst, and then went to medical school at Trinity College in Dublin, graduating in 2017, and did an internship in both Galway and Letterkenny University hospitals prior to coming to join our residency program here at BBCH. Dr. UTLEY has pursued research in hematology oncology in particular, looking at APC as a marker of bone marrow recovery, looking at the impact on holding stress and bonding in mother-infant pairs during therapeutic hypothermia with Dr. Alexa Craig. She helped to revise the fever and neutropenia guidelines, and she has several publications related to her research. She has a number of awards, including the Medical Final Prize that she received at Trinity College in Dublin. She was also a nominee for distinction for the Pathology Award. She's a volunteer here on the Wellness Committee, Maine Families for Vaccine, voting, encouraging families to vote no on the vaccine to prohibit the vaccine refusal issue back in the spring. And she is going to be talking to us today about CAR T cell therapy, a living drug. Dr. UTLEY matched successfully into an oncology fellowship program at the University of Colorado, and we are thrilled to listen to her grand rounds today.
Good morning. Thank you for that very kind introduction, Dr. O'Leary. So as she mentioned, I'm KAYLYN. It's nice to see. I have a ton of people actually here in Kalum Family Tower with me today, which is unexpected. Just as a heads up for you guys, the sound from this is coming from my laptop. So if you decide to gather within socially distanced guidelines, that's fine with me. So there's your CME text code. Let's see if I can get this. There we go. So again, thank you all for being here. Also, thank you to my grandmother who is probably in the audience somewhere. So everyone be on your best behavior. She's very excited for this. I'm going to be talking about something that's really fascinating to me, and I think it's incredibly cool therapy emerging in pediatrics, especially in oncology, but really pediatrics broadly. And this is chimeric antigen receptor T cell therapy, which I'm going to refer to as CAR T cell therapy, mostly in its abbreviated form. Alrighty. So I have no disclosures. I will be no richer after I give this presentation. All right. So just to review some objectives with everyone, today I'd like to discuss the evolution of therapies in oncology. I'll briefly review some basic T cell physiology, and I'll also define CAR T cell therapy and talk about how it works. I will be reviewing some of the data behind CAR T cell therapy in relapsed and refractory B cell ALL. I'll also describe some of the associated adverse events or adverse effects and mechanisms of CAR T cell failure. And I'd like to also discuss some of the future applications of CAR T cell therapy and similar technologies. And I'm going to put you guys down here. Okay. All right, so this is the best slide. Many of us have seen this at some point or another in our careers, and this demonstrates the overall survival among children with acute lymphoblastic leukemia since 1968. And so if you look down here, I hope you can see my cursor, yep. Starting at 1968, when survival was near 10% for these children, there have been massive strides overall, which is due to the implementation of steroids and then the addition of intrathecal chemotherapy in the early 70s. And really from there, it's been tweaking the schedule and doses of a lot of therapies rather than the addition of many new therapies. This has effectively brought overall survival close to 90% for these children, which is really staggering. But it's important to kind of think about how the landscape of cancer therapy is changing now. And that brings me to the discussion of precision medicine and targeted therapies. So newer discoveries about the genetic basis of a lot of cancers has led to the development of genetic, epigenetic, proteomic targeted therapies, as well as huge advancements in immunotherapy. And these directed therapies really hinge on the concept that they're specific to the patient compared to traditional chemotherapy, which acts more broadly on just dividing cells. So to talk about targeted molecular therapy, I have to mention probably one of the biggest landmark achievements in personalized oncology, which is the development of Gleevec or imatinib for CML. This drug came to the market as a revolutionary therapy for patients with CML in the 90s, decades after the discovery of the famous Philadelphia chromosome, which is this tiny, puny little chromosome 22 over here. This drug specifically targets the tyrosine kinase mutation that causes CML, as well as some other types of malignancies, including some ALL. Other targeted therapies, though, include those directed towards hormone receptors, angiogenesis, gene expression, as well as apoptosis. And immunotherapy is another example of precision medicine that has really been in the forefront of oncology over the last decade. Simply put, immunotherapies, they harness the innate immune system to kill cancer cells, and they include a ton of different approaches, including monoclonal antibodies, immunomodulators, checkpoint inhibitors, and cell-based immunotherapies like CAR T-cell therapy. So just one example is shown in this little graphic here. It shows the PD-L1, PD-1 pathway, which I kind of think of as an immune system emergency brake. Some cancers will use this to turn off T cells that are targeting the cancer themselves. And the checkpoint inhibitors can come in and flip this switch, which is shown on the bottom, empowering the T cells to then kill the cancer cells again. And they've been really helpful in a variety of cancers like melanoma, Hodgkin lymphoma, and lung cancer as well. So I wanted to give you guys a little bit of history. So we'll take things all the way back to 1891. This man is William Cooley. He is a bone sarcoma surgeon working in the US, and he noticed that one of his patients had a severe skin infection, and then he was cured of his disease. So he performed the first study of immunotherapy for the treatment of a malignant bone tumor, and this is absolutely bananas. So he injected organisms, streptococcal organisms, into a cancer patient to cause erysipelas and stimulate this patient's immune system. This patient's tumor disappeared completely. Unfortunately, the subsequent 2 patients who he did this to died from the experiment, prompting him to use a heat-killed organism, which later became known as Cooley's toxin. By the end of his career, he had treated nearly 1,000 patients with this toxin, with reports showing as many as 500 achieving near complete regression of his disease. Though I will add that his daughter was the primary researcher to take over analyzing this data, so there is room for bias here. But this technology fell out of favor due to cynicism, as well as the advent of modern chemotherapy and radiotherapy. However, in more recent years, scientists have returned to this concept of really manipulating the immune system to heighten response to the disease. And with all of this background, a lot of people refer to him as the father of immunology or the father of immunotherapy. So I'd like to get into some really basic T-cell physiology because I had to do this myself when trying to understand this therapy. So to start with the basics, if you guys take it all the way back to med school, we can talk briefly about T cells. Their precursors are made in the bone marrow. They travel to the thymus, where they then differentiate into 2 major types of cells, the CD4 helper T cells and the CD8 cytotoxic T cells. The helper T cells really help enhance inflammation. They signal other cells to get to work. So that's this one here, sending out all of its cytokines. And the cytotoxic T cells are killers. The image shows here a virally infected white blood cell, but the principle is exactly the same for cancer cells. And I'll note that some of these CD4 and CD8 cells can go on to differentiate into a variety of different T cells as well. So what makes them suitable for harnessing as tools in oncology? There's really a number of reasons for this. As part of the adaptive immune system, and this is a T cell just absolutely destroying this cancer cell here. They're targeted, they know exactly what they're looking for, and they hunt it. Searching, they can kill hundreds to thousands of cells every day, and each cell is capable of multiplying 10 to 100,000 times. They only live for a couple of weeks, and they can travel anywhere. This includes the skin, the CNS, and obviously they can circulate in the blood and lymphatic system as well. So if these cells are designed to kill cancer cells, why do we get cancer in the first place? There are a few tricky things that cancer does to trick our T cells, and this is just a representation of a few of the major tools that cancer uses. So another throwback is going back to the major histocompatibility complex. This serves to present antigens on cells, whether they're self or non-self, and they present them to T cells. Tumor cells, very cleverly, can downregulate the expression of MHC, making it harder for the T cells and immune system to see and recognize these cells as abnormal. Cancer cells also produce abnormal antigens themselves that prevent our T cells from attaching, whether they're just extremely abnormal or longer, making it difficult to reach. And they recruit their own immunosuppressive team of cells and signals as well. And lastly, I have here, they can manipulate immune checkpoints, which is kind of what I was showing in that cartoon in my previous slide. So finally, this brings me to CAR T cell therapy. CAR T cell therapy restores the T cell's ability to recognize and destroy the cancer cells. That's really the science behind it. So the chimeric antigen receptor, which is shown here, this bracketed area, this is incorporated onto the surface of T cells, typically immune or immature cytotoxic and helper T cells. And once they reach their target, which is this tumor cell here expressing a specific antigen, they're activated killers. Just as innate T cells would, they multiply, they recruit others, and they kill. And their ability to kill and expand like this is what tags them as a living drug. They also function independent of MHC molecules completely, and they bypass chemo resistance, cytogenetics, and other patient factors that really make broader therapies quite challenging. And something that's even cooler about CAR T is that while these populations of T cells are alive and persisting in the body, they offer surveillance. They are constantly moving through circulation, hungry for their exact antigen that they're looking for. So how does one select the right antigen to target? And one of the challenges with CAR T is that you need to target an antigen that is specific to the cancer cells, but is not present on normal cells. And by far, the most studied CAR is engineered to target CD19. CD19 is a marker that's ubiquitously expressed in the B cell lineage specifically. So if you look at this image over here to the right, you can see these CD19 antigens that are, or antibodies that are expressed in everything from the early B cells all the way down to the plasma cells. And that makes them really, really useful for a lot of B cell malignancies, which are abbreviated here, including B cell ALL, diffuse large B cell lymphoma, and mantle cell lymphoma. And the real question, though, beyond this theory is, can this actually work? Okay. So you can see here, the first use of CAR T cells were they were developed in Israel in the 1990s. And the first use was in, sorry, I'm just looking for my notes here. Here we go. Is in mice injected with Burkitt's lymphoma, which is a cancer that's ubiquitously expresses CD19. They were completely immunodeplete mice who received these CD19 CAR T cells. So they had no immune system, and they received the human CAR Ts. And if you've never seen a PET scan of a mouse, this is what it looks like here on the left. So you can check that off your bucket list. These images are both highlighting the typical sites of FDG uptake in a PET scan, like the brain, kidneys, bladder, and heart. So brain, heart, kidneys, bladder here. And the image to the left is that of an untreated mouse who has lymphoma. And you can see there's this uptake here, both in the vertebral column and the calvarium, which are the bones of the skull. And the image to the right is a mouse 6 months post-treatment with CAR T cells. This image shows that you can see there's no uptake of the vertebral column anymore or the calvarium, which shows a dramatic improvement. And this image over here also shows the dramatic improvement in survival of these mice after a single infusion of this therapy, even persisting up to 300 days later. What does the process of CAR T actually look like for a patient? So first, the patient's blood cells are removed via leukapheresis. The patient's blood goes into a special apheresis machine, which is the same machine that's used for autologous and allogeneic stem cell transplantation. Blood goes out, cells are extracted, and blood is returned to the patient. The extracted cells are then sent to manufacturing, so that's shown here in number 2. They're transduced with a viral vector. Usually it's an inactivated lentivirus or retrovirus. It's helping with this. And they carry the genetic code for the CAR itself. So the cells are then expanded to the lab, which is shown here, and they're, before returning these enhanced cells back to the patient, first the patients are given lymphodepleting or reducing chemotherapy, which really serves to make room for this new immune system. And then once this is all done, the T cells are infused, and the process typically takes a few weeks in total. And this here is an image that shows just that, CAR T cells working in a patient. So this just shows glioblastoma cells. And if you just give it a second, it'll kind of recycle. And so here's the glioblastoma cells. And here you can see these CAR Ts coming in, recruiting each other and kind of engulfing this tumor with these carcinogenic cells. Oops. All right. Alrighty. So I'd like to talk about B-cell ALL as really kind of, this has been the direction that CAR T started in. ALL, as I mentioned, it's the most common type of pediatric cancer, with B-cell ALL comprising about 85% of cases. While overall survival is great, as depicted by my earlier slide, unfortunately about 15 to 20% of patients do relapse. In fact, relapsed B-cell ALL alone is the 4th most common pediatric cancer and remains a leading cause of death from childhood cancer. Patients that are refractory to induction therapy have an estimated event-free survival of about 32%, compared to 77% in patients who are disease negative after their induction, so their first part of their chemo. Survival in the setting of relapse relies heavily on its timing and the location of the relapse disease, and survival worsens after every subsequent relapse. Survival in relapsed and refractory disease, which is stratified by risk, is what is shown in these figures here. So on the left is survival probability for relapse within the bone marrow alone, and you can see, so this is IMR, is isolated marrow relapse, and this is stratified based on the timing, and you can clearly see that the earlier the relapse, the worse the outcome. And then this right image shows concurrent marrow relapse, so CMR, meaning that there's disease elsewhere, like in the serum or in the periphery or in sanctuary sites. And you can see the same trend as well. The earlier the relapse, the worse the outcome. Stem cell therapy can offer durable remission for some patients, especially in first relapse, though it carries tremendous risk, high morbidity, and few options are available should this fail. Other promising therapies, including targeted antibody therapy to CD19, which is called blinatumomab, for relapsed and refractory B-cell ALL, have shown some promise, with response rates, though, only up to 40%. And the high rate of relapse is associated as well, with a median survival just shy of 8 months. So you can see CAR T is a promising new direction for therapy, providing a favorable option to a lot of these patients who have pretty poor prognosis. So I wanted to show you a patient because the big question here is, can this actually work in humans? So this is the story of Emily Whitehead, who was the first child in the US who was enrolled on a CAR T study in 2012. And this might be a little tricky for you guys in the room to hear, but hopefully it's loud enough.
Blessing to us. My name is Emily. My favorite color is purple and pink and bluish greenish. She's just a really creative little girl. She loves to draw and paint and do crafts. We think that she's just really strong and really brave because of what she went through. She went through 16 months of chemotherapy. She was supposed to go through a total of 26 months, and we got to 16 months of treatment when we found out that she had relapsed. The chemotherapy didn't work. And our hospital there told us that there wasn't anything else that they could do. And so they said that we could take her home and we would have a few weeks with her, or possibly a few months.
It even got to a point where Carrie had to stop doing research because the research showed that she wasn't going to survive. So, I would tell her, no one has done a study on Emily yet. It's not an option not to have a positive outcome here. And it's going to happen.
Emily came to us in April of 2012. She had been in the hospital for nearly a year and was unable to get into remission. She was the first child enrolled on our protocol. So we had a lot of doubts as whether the infused killer T cells that we made would be able to keep up with the rapidly progressing leukemia cells.
Some parents naturally would think of, I'm not sure if I just want my child to be a scientific experiment, but trying these experimental studies is what leads to breakthroughs.
I had read that immunotherapy was what doctors and researchers were kind of going towards, to use your immune system to fight cancer. The way that they explained it to us in kind of simple terms was that they would take her T cells out of her blood and that they would train them in a lab, or that they would send them to boot camp. Then we're going to put them back into your blood, and those cells are going to find cancer cells in your body, and you're going to get rid of them. They had expected her to get a fever about 7 to 10 days after the T cells. She was on a ventilator for 2 weeks. But once she got on the ventilator, I think the doctors and researchers realized that it was because the T cells were actually working.
And my cell phone rang.
He looked like he was in a daze, and he was just staring at his phone. And I said, "What's wrong? There must be something wrong." And he said, "No." He said, "Dr. Grupp just called me, and he said the T cells worked." He said she's in remission.
It's been an amazing journey for me. It's been over 20 years that I've been working on this, so that's obviously very rewarding. There's still a lot to do.
Okay. So as you can see, it's a pretty incredible story and quite a risk that was taken. And this is now, I think this video is a few years old, and now 8 years later, she is still in remission, which is great. Oops. Okay. So after Emily's success, along came a trial. This is out of CHOP, looking at a total of 25 patients who were under the age of 22, and 5 older patients as well, 26 of whom had relapsed B-cell ALL between their first and their 4th relapse, and 3 had primary refractory disease. 1, interestingly, had T-cell ALL that happened to express CD19. And the data from this study was pretty remarkable. 90% of patients were in complete remission 1 month after their infusion of CAR T. There were 2 patients as well who had blasts in their CSF at the time of the infusion, and there was no detectable CNS leukemia at their 6-month follow-up, and no CNS relapses were observed. At 1 year, 63% had achieved sustained remission, so they had event-free survival, as I'll kind of keep referring to, with an overall survival at 1 year of 78%, which is pretty staggering when you think about the numbers that we were looking at before. And then a follow-up trial came from this. So the second study, this is a phase 2 multi-center randomized international trial This is for children and young adults with relapsed or refractory B-cell disease. And the CAR T was given this name here, and I had to practice saying this like at least 40 times. I still don't get it right, and I found this YouTube video that will say it for me. Okay, so tisagenlecleucel is a manufactured CAR that I'll touch on again later if I can say it then. The study was conducted at 25 different centers and included 75 patients ages 3 to 21 with relapsed or refractory B-cell ALL. And notably, about 2/3 of these patients had experienced relapse after stem cell transplantation as well. The study showed an overall remission rate of 81%, and remission was durable, with 80% achieving relapse-free survival at 6 months. And the overall survival at 6 months was 90% and 76% at 12 months following infusion. So you can see that even a single dose of CAR T-cell therapy can produce high rates of remission and durable remission as well. All right, so another thing that we learned from these studies was about the adverse effects associated. So the first and probably the most significant one that I'd like to cover is cytokine release syndrome. So occurring in about 70 to 80% of patients, this is a systemic inflammatory response. Once in the body, the CAR T cells, they activate, they proliferate, and they cause target cell lysis, all of which will induce an explosion of cytokine release. And so this figure is kind of helpful in thinking about the other cells that are also included in this massive storm of cytokines. With an understanding of the physiology, it really is almost an expected phenomenon, and it exists on a spectrum. So you can have mild and self-limiting CRS, which can be fever and myalgia, much like you'd feel after your vaccine, or to severe and life-threatening, which can include vascular leak, hypotension, respiratory and renal insufficiency, and coagulopathy, and even, unfortunately, sometimes death. But the majority of patients experience the mild to moderate variant of CRS. And there are several factors that come into play when predicting the risk and severity of cytokine release syndrome. So simply put, patients with more CAR T cell expansion seem to be the ones who have more severe CRS. So this means patients who have a higher tumor burden at the initiation of therapy, and those who are more lymphodeplete prior to initiation of treatment are at higher risk for severe disease. So when patients are lymphodeplete, it really leaves room for the CAR Ts to expand without any mitigating factors of the innate immune system. So there's absolutely no dampening, and that's why being lymphodeplete before the treatment makes you a little bit more at risk for the severe CRS. So patients with more severe CRS, as depicted by the red lines in these graphs, tended to have earlier fevers. So you can see this in figure A, which shows kind of the timing of the fever itself. And you can see the earlier the fever here is really associated with the higher grade CRS. It also, these patients, their fevers tended to be higher, so greater than or equal to 38.9, which you can see here in figure B. Again, the red line is the patients with the more severe CRS, and you can see their fevers are statistically quite higher. And these patients also had much higher inflammatory markers really across the board, which is shown in C and D. These are just two of many inflammatory markers that have been looked at, all of which seem to be elevated and help just demonstrate that trend. An important question here too is how is it managed? And since the goal of CAR T cell therapy really is to induce in the activation and expansion of CAR T cells, dose reduction is not the ideal option in order to prevent this. So really it's helpful to identify these factors early on and then monitor these patients closely in the appropriate setting. Severe cytokine release syndrome can often be successfully managed with an anti-IL-6 receptor antibody called tocilizumab, with or without corticosteroids. And the tocilizumab often results in rapid defervescence and blood pressure stabilization. Some patients will receive steroids and others will receive a second dose of tocilizumab. And interestingly, it doesn't seem that offering these medications interferes with the effects of the CAR therapy at all, which was an initial concern when offering this kind of rescue medication. And another important side effect to talk about is neurotoxicity. And it's not an uncommon phenomenon after CAR T infusion. It happens in about 40 to 50% of patients, and symptoms range from delirium to hallucinations to fatal cerebral edema. The presence and the severity of the CRS is the greatest predictor for neurotoxicity. So it's shown in the graphs here. The left shows higher grades of neurotoxicity, which are these darker colors. Sorry, you can't really see this. I'll move you again. So these darker colors here are associated with the higher grade of cytokine release syndrome over here. And then the image on the right shows the correlation between high fever and the development of neurotoxicity. Okay, you can see that there. And these events occur sequentially as well, which supports interesting evidence that the systemic inflammation during CAR T actually leads to a disruption in the blood-brain barrier and ultimately leads to astrocyte activation and trafficking of other immune cells into the CNS. And interestingly, trafficking of the CAR T cells themselves and having CNS disease doesn't seem to correlate with the neurotoxicity. It really is very dependent on the degree of the cytokine release. These images at the top show some of the many patterns of signal abnormality appreciated in CAR T-specific neurotoxicity. A and B up there show non-specific patchy white matter hyperintensities. C shows brainstem hyperintensities, and D shows bilateral thalami hyperintensities. So again, they're really non-specific, but there are a number of patterns that are seen. And management of this neurotoxicity is mainly supportive with a focus on seizure prevention and attenuation of the immune system, often using corticosteroids. It also is important to highlight that this is typically a self-limiting phenomenon, though it may necessitate ICU admission. And then another commonly appreciated consequence of CAR T is B cell depletion, which is really an anticipated effect. So as CD19 is present on these B cells and their precursors, as we already mentioned, you expect this phenomenon when CAR Ts are in circulation. And the big issue here is hypogammaglobulinemia. The B cell aplasia really serves as a marker of CAR T function, and it occurs in every single patient who has a response to therapy. And as we mentioned before, B cells are considered non-essential, and the aplasia is actually well tolerated. It persists as long as the CAR Ts remain in circulation. And the hypogammaglobulinemia, while it does put patients at higher risk of infection, is treated effectively with IVIG. And as I mentioned already, it's pretty well tolerated. So indications for CAR T cell therapy, there are 3 FDA-approved CD19-directed CAR T products which are available in the market. So the first is the tisagenlecleucel. I'm so pleased I just said that. It turns out there are a lot of ways to pronounce that. So Kymriah is what it's called. And this is licensed for B-cell precursor ALL that's refractory or in its second relapse or later, as well as adults who are, have relapsed and refractory diffuse large B-cell lymphoma. This will cost you up to $475,000 for a single dose, which is a massive amount of money. I did see more recent quotes that were looking at closer to like $375,000, which could be related to the fact that more products are coming out. There may be a little bit more competition in the market. But if you're kind of freaking out a little bit about the cost, it is similar to the cost of a bone marrow transplant as well. And a couple other therapies that are available are Yescarta, which is licensed for diffuse large B-cell lymphoma, primary mediastinal B-cell lymphoma, high-grade B-cell lymphoma, as well as diffuse large B-cell lymphoma that results from follicular lymphoma. And then there's also Tecartus, which is for relapsed or refractory mantle cell lymphoma. This is the newest one that's been approved. All right. So as we had already mentioned, CAR T can achieve a high remission rate, but it is still associated with a relatively high rate of relapse. And I think it's important to understand why and how this happens. Crudely, this can be broken up into CD19 positive and CD19 negative relapse. So first, It's expected that the CAR T population generally will die out eventually as marrow precursors will replace the population. However, there are some memory T cells that will persist, hopefully. However, for the therapy to work, they really have to outlive the tumor population. And they also, you have to think about them overcoming some tumors that have like a massive expansion and doubling time. So in the realm of CD19 positive relapse, there are a few different things that can happen. T cells can be exhausted, and this is a specific stage of T cell differentiation caused by repeated antigen exposure, which weakens the function of the effector T cells. And then there's T cell senescence, which is a little bit different. It's considered irreversible, and it's really mainly due to age and telomere shortening. And there's also immune escape mechanisms. So again, we come back to the tumor manipulating its local environment. Then when you're considering CD19 negative disease, some of these patients actually may have a small population of CD19 negative blasts prior to CAR T treatment, which means that the CAR T cells are effective where they're going, but these other blasts can live on and become the dominant clones. And then some patients will experience a lineage switch from a lymphoid to a myeloid malignancy. So as you can see, CAR T is really becoming a widely accepted treatment modality in the realm of B-cell malignancies. Why aren't we trying this on every single malignancy and beyond? Well, there's a lot of reasons actually. And few of the reasons that account for the success of the CAR T in B-cell disease we've already talked about. One is there's an antigen. Clearly there's something we can target. And another is that we can afford to lose all of the cells with that antigen, as long as you have IVIG replacement. So I wanted to talk specifically about some of the challenges of applying CAR T to other malignancies and a little bit about what is being done to overcome this. I also want to show you some of the exciting developments in CAR T technology outside of the realm of oncology. So let's talk about liquid malignancies first, and I'll start with AML because it's another malignancy that we've really all encountered at some point in our training. We definitely see this on the floor. Relapse after conventional chemotherapy is a huge challenge in AML, and currently stem cell transplant is the only curative option after relapse. As we've seen, CAR T has offered safe and durable remission for ALL, so why not AML? And the particular challenge here is that what myeloid malignancies seem to be lacking is a specific dispensable antigen. So what I mean by that is the antigens on AML are typically co-expressed on normal myeloid cells and the hematopoietic stem cells. So you can see that here. I've kind of written some of these antigens next to the myeloblast where AML kind of occurs. You can see CD123 and CD33 are both present on the common myeloid progenitor. And you can see CD123 present as well on the multi-potential hematopoietic stem cell. And targeting these would lead to intolerable marrow ablation. So there's some strategies to overcome this problem, which are pretty fascinating. And the first is implementing a safety switch that allows the T cells to be turned off. So that means that once you've achieved the response that you'd like, which is that you're in remission, you could have an emergency switch on these T cells to stop future myeloablation, as long as you get there ahead of time. Another option is engrafting them with stem cells that have been engineered without these same surface antigens. So therefore, the engrafted stem cells would be resistant to the CAR Ts, ensuring that you could just continue your therapy as normal and you wouldn't have marrow ablation. But really, the holy grail of this is going to be finding that neoantigen of AML to help differentiate abnormal blasts from normal cells. And there's extensive genetic analysis of AML underway to help do this. And another cancer that's worth talking about is multiple myeloma, which, yes, we do not see much of in the pediatric world. But this is pretty cool too. So CAR T cells targeting B cell maturation antigen, so BCMA, that's shown over here in the plasma cell, have been investigated in a number of studies and have demonstrated important disease remitting activity. Its expression increases during B cell differentiation, which is also super helpful. So it only is found on late memory B cells and plasma cells. And moreover, malignant plasma cells seem to be expressing higher BCMA levels than normal plasma cells. In trials of patients who have relapsed after at least 3 lines of therapy, including proteasome inhibitors, immunomodulators, other antibodies, The observed response rate exceeded 85% in these patients. And some enrolled patients in one study who responded to this directed CAR T had failed as many as 8 other prior therapeutic regimens. And this image here is testament to that. So this shows on the left before and on the right after. These are PET scans of a patient with multiple myeloma who was treated with a single dose of the BCMA targeted CAR T cells. So another challenging area that's definitely worth discussing is that of solid tumors. And the challenges here are many. So first, there may be a lack of T cell migration to the tumor site itself. And the tumor microenvironment within solid tumors is notoriously immunosuppressive, coming up with very clever ways to dampen the immune system. And in these malignancies, it's proven very difficult to find a specific antigen that targets tumor cells and leaves the normal parenchyma alone. Cannot simply target the parenchyma of the lung, the liver, or brain without catastrophic deficit. There are some early and ongoing trials that have shined a light on ways to address some of these. And the first I want to mention is shown here, which is high-risk neuroblastoma. And as a lot of us are familiar with, this disease involves the kitchen sink approach to treatment, which carries a high morbidity. And despite aggressive therapy, long-term survival is only 50 to 60%. So this really does make it an interesting and promising target for CAR T. The target antigen in the study is GD2 surface protein, which is also the target antigen of the monoclonal antibody dinutuximab, which some of us have seen given on the floor as well. Children treated with this showed a rapid reduction in tumor size, although its anti-tumor activity was really transient. But there are many, many studies underway examining neuroblastoma specifically and the role of CAR T. And then currently there's a phase 1 trial enrolling children with relapsed or refractory HER2 positive pediatric central nervous system tumors. This is also wild. So using local regional therapy, meaning that the CAR Ts themselves are injected right into the resection cavity or into the ventricular system itself, researchers are hoping that those CAR Ts will be safe and effective. And the same question is really being asked in a similar phase 1 trial for children with diffuse intrinsic pontanglioma, which is notoriously a horrible prognosis, and other refractory CNS tumors using a different antigen, which is B7H3. One essential finding from all of these studies that we've talked about is that in order to be effective in killing cancer cells, the CAR T cells need to expand and persist in the body for prolonged periods. And for both of these to occur, you really need adequate co-stimulation of the CAR T cells. This has led to numerous modifications of the original CARs. So I'm going to talk about 4 generations of CARs. The first generation here on the left had only this one signaling domain, which is shown here. And that was proven to be inadequate. It failed to persist in vivo more than a couple of weeks, and so didn't really do much. So then came the second generation CAR, which has another co-stimulatory but cytoplasmic domain. And this is most commonly used in generations of B-cell malignancies. So that's what's in Tysogen Laclusil and the other licensed products. And then you have these third generations, which have another co-stimulatory domain. And then this fourth generation, which is really cool. These are called trucks, and they offer targeted delivery of cytokines. So these are used more specifically in the setting of solid tumors, which I mentioned are notorious for having a really, really immunosuppressive microenvironment. So they can be quite useful. But for everyone who's not as excited about oncology as I am and maybe half asleep, I want to talk a little bit about the applications beyond oncology. So one of the earliest CARs to enter investigation was the use of CAR Ts directed towards the extracellular envelope protein for HIV on CD4 cells. And this really has been a success in terms of learning a lot from CAR T, but has been a failure in terms of inducing complete cure of HIV because it's failed to eliminate latently infected cells. But there are a lot of efforts to overcome this roadblock. CAR T cells have also been used to eliminate autoreactive T cells in an early study of animal autoimmune encephalitis, which serves as a model for multiple sclerosis. And then more recently, autoantigen-based immune receptors, which are named CAR T cells, but with 2 A's, they were developed to eradicate the self-reactive B cell clones So that's shown in this image here. These are the self-reactive B cell clones. And these target the loss of keratinocyte adhesion in the autoimmune disease pemphigus vulgaris. So this shows some of the outcomes after this treatment. You can see the derangement of the keratin layer up here, and then a patient after CAR T cell therapy, you can see their keratin layer is back intact and there was not any skin toxicity, which has been pretty remarkable since therapy is very challenging in that disease. And another similar and clever strategy is to apply the CAR technology to regulatory T cells. So these are further differentiated T cells from the original ones we mentioned. They have immunosuppressive rather than cytotoxic function. So this approach has been evolving in the setting of autoimmune disease. A regulatory T cell helps dampen inflammation and can also be genetically manipulated by retroviral gene transfer, as I mentioned. So they're then guided by the CAR in a specific manner, targeting a cell of interest and expanding to halt inflammation. And the data for this has been really, really exciting. So there's been some promise in enhancing allotolerance in solid organ transplantation, suppressing inhibitory antibody development in hemophilia, and curing colitis in murine models, which is seen here. So this is a study where they took transgenic mice who were transplanted with CAR T cells, regulatory cells though. And these mice are shown in these little black squares here. And the CAR in this case was directed towards specific colitis-associated antigen, and these mice were completely cured of their colitis. So if you look at the wild type here is these little triangles, you can see the survival percentage is significantly different. And this is a really cool histologic picture showing the massive amounts of inflammation in the wild type mice compared to the mice that were treated with the T regulatory cells. So there's a lot of attention on this in the last 10 years, and I hope you can see why. It really is something that I think is very exciting and will come to play in every single subspecialty moving forward. And just to wrap up with some conclusions for you guys. So CAR Ts, they are personalized and targeted therapy, and they have the potential to overcome means of drug resistance that are adopted by many, many cancers. They've offered effective and durable remission in patients with relapsed and refractory B-cell ALL. The adverse events, as we talked about, can be serious, though they're generally tolerable. Cellular therapies, including CAR T, are currently being developed and trialed for many, many diseases. So both in the realm of oncology and outside of oncology as well. And lastly, I just want to say thank you. Thank you to Nadav Castle, who was incredibly helpful in putting this presentation. And thank you to Say Z, who is just my be-all, end-all mentor and always available to me. Thank you to Tom Reynolds and Carol McCarthy for helping me put this presentation together as well. And thank you to my spectacularly amazing co-residents. I adore and admire all of you. I would not be where I am if it wasn't for all of you guys. All right, there's some references, just a few. And yeah, now would be a great time to take some questions. And I think I can help navigate this. There is a tiny applause coming from this room, if anyone's wondering. Thanks, guys. All right. Yeah.
So your goal is to make memory T cells.
Mm-hmm.
Using the memory T cells against CD19.
Yeah.
So are these kids or people who get these, are they going to be immunosuppressed? Everything like that?
That's a good question. So the question was, the goal is to really get to the point of a memory T cell. And are these kids going to be immunosuppressed long term? So we do have real oncologists on the phone if anybody wants to chime in, but I will answer this as best I can. So the cells that are actually going in are going to be the immature cells first. And those are going to be targeting, they're creating that massive cytokine storm. They're the ones targeting. You hope that you eventually end up with some memory T cells, but they're not going to cause immunosuppression necessarily long-term as far as I'm aware. But as long as there's active T cells that are working and are in number enough to overcome the cancer, they'll be working. I don't know if anyone else has anything to add, but there have been different technologies to try to make sure that you get those long-term memory T cells too. I can look at some of these questions in here. what are the circulating half-life of these modified T cells versus endogenous T cells? That's a great question. It's really, really patient dependent. And it also depends on the generation of the CAR T, as I mentioned. So as long as the CARs are present, the patient should be in remission, and obviously after some point. But so for example, Emily, the patient that we had talked about, she still has these cells in circulation. So they can last for a really, really long time, but they can be short-lived as well. So it's very variable and patient dependent. I don't think it's really understood quite yet. And then gastroenterology wants to know what colitis means. It's an animal model of colitis that's supposed to be similar to both UC and Crohn's. So it's kind of a crude measure, but it is more of a specific inflammatory response rather than just like a local colitis. Oh yeah, and there's the CME code. Any other questions or comments from you guys? George, well done. Thanks, George. So we're done early, which never happens. All right, well, thank you all so much for coming. I hope you were able to stay with me for a lot of that and got excited about this stuff too.
Yeah, thank you very much, KAYLYN. I think you did an excellent job explaining the pathophysiology in a way that I've heard talks on this multiple times, but I think this was the clearest and the most straightforward talk about the applications of the CAR T therapy. It's really exciting. So thank you so much for giving us such an excellent overview of this new therapy.
Thank you. All right, there are a few other questions actually here in the chat that I had missed. Okay, let's see. Michael said, let's see. Oh, he worked with some of the first cases of CAR T and worked with Emily Whitehead at CHOP. That's cool. And he's reiterating that you can't stress how sick they get. I think everyone can see this because I'm still sharing my screen. Fevers 107, SIRS, need 2 to 3 inotropes and pressors. Yeah, they're really, really sick. But these are kids that had a 0% chance of cure. Okay, long-term follow-up on neurotoxicity. Oh, that's a great question. I think it's individual based. There are very, very few patients who have long-term neuro deficit after this. So most, like I said, most of the time these are self-resolving events. And I think just the numbers are not there to make it like a formal recommendation on neuro follow-up. There is question about whether all patients should have MRI imaging before the study so that they can know if these changes are acute versus, or sorry, before the T-cell therapy, so that people can know if the changes are acute versus chronic. And it's not uncommon to see chronic changes that worsen because of the CAR T, which is interesting too. I think, okay, I think that is all. Thanks, guys. All right.